Before understanding how the HRAS GTPase protein relays signals from outside the cell to inside, you must first understand the simpler components about it. For instance, here I've shown a GTP molecule. As you can see, I've shown three tail-like structures which are called phosphates. Essentially, when a molecule called GAP goes and snips one of these phosphates away, it results in another structure, and the new structure's name is called GDP. As you can see, it's the exact same as GTP, except one of the tails is missing due to the gap snipping function. Now that we have understood how GTP and GDP relate with each other, we can relate both of them to HRAS's function. So, as you can see, it's listed here. When GTP is bound to HRAS, it's considered switched on. And to switch it off, use the function of gap, as we discussed before, to change a GTP into a GDP, ultimately switching off the whole protein. Now, we actually haven't talked about GEF before, but essentially it does the opposite function of gap, and it helps the GDP change into the GTP, switching on the protein. And this is essentially how the HRAS GTPase protein relays a signal, by constantly switching between the GDP off version and the GTP on version. But what is the protein actually achieving by switching on and off? Well, when it's switched on, it also switches on other proteins. And this in the end will eventually result in a signal for cells to either grow or divide in the human body.